So I'm here today, I think for the third or fourth time in a row, but my first time as a member of the Regeneron Genetics Center, which I've joined in February. I'm doing a very similar gig, except I don't have to write grants anymore. But one of the main differences between, I think, what I did at Baylor College of Medicine at the HGSC, working with DNAnexus, doing large-scale sequencing, and what I'm doing at the Regeneron Genetics Center, besides the purpose of doing that, is that both places were doing at-scale genomics. But what really struck me as soon as I showed up was the idea of doing at-scale genomics for at-speed discovery. We're moving really fast. So that's what I want to sort of walk through today and sort of give my experience with some of the differences between what I did in academia and what I'm doing here in industry, and how that has sort of changed the type of informatics infrastructure that we're looking at. So when I think about the path from genomics to precision health, in academia I was very much in that column over on your left. Where this idea of genomics, and there's all these complicated, intricate steps that are involved in collecting samples, getting the consents, doing the sequencing, doing the analysis with the phenotypes. You know, these are people with dozens of years of experience pouring their, lives and careers, ruining their lives and careers into these areas. And you do all of this work, and a lot of time in academia, what that ends up with is a publication. And it's great. You get this publication, and you hope that someone else sort of takes it to the next step. Now, there are opportunities to take what you find in these papers, which are association tests or polygenic risk scores or things along those lines, and you want to link that to precision health. You want to link that to trying to improve the health of individuals or populations. And a lot of the ways that we use that in academia is decision support. Whether it be, can we improve a diagnostic? Can we assess genetic risk that might impact lifestyle? Can we think about what treatments to give? Can we modify the behavior of patients? Can we inform physicians? There's all this stuff that goes on, in people's heads. But it's not like we find an association and all of a sudden, someone's just automatically healthier. There's all these other steps that have to happen. Now that I work with a drug company, we make drugs, right? We're trying to find things that we're trying to find pills that maybe while they won't zombie your brain, they will improve your health in some way. And that's, one of the differences is this sort of direct effect between what we discover with genomics and the phenotype data and impacting, human health and disease. And the Regeneron Genetics Center sort of made this commitment. We just had our 5-year anniversary about a month ago. I get to say we now, not them, because I get yelled at. But the idea is that you cannot do modern drug discovery without incorporating human genetics. And I think Marilyn made a lot of really nice points about how the discovery aspect of this can really drive this target identification. These are just a couple of summary numbers that explain that this process, the clinical trials, and all of these, the R&D that goes on, the cost and the time, and most importantly, the latency between, hey, we think we have a scientific discovery. We think we can explain risk. We think we can explain this disease genetically. Okay, that's great. It gets an article in, whatever newspaper you read, and everyone gets excited, and they're like, my grandfather's sick. When are you going to help him? Well, give us 10 to 15 years, and, we'll have something through a trial, maybe take something. Right? We want to accelerate that process. And a lot of the things that you hear about at a lot of these precision medicine conferences is about accelerating the analysis of the data. How do we go faster from these discoveries to something actionable? I'm going to step back because I live in production land. I sift through genomic data, and I worry about tiny problems that turn into big ones. On behalf of our collaborators, okay? So the RGC has a network of 70 plus collaborators. This is an old slide. It's probably much higher than that now. And a lot of samples, that number is definitely lower than it should be. But I haven't stolen the new numbers yet. These are just some of the collaborators that we work with, we sequence on behalf of. Obviously, the big ones are the UK Biobank. We just had our first public release of data in March, the 50K samples, our sort of foundational collaboration with Geisinger. But it's not just large, cohorts that we're sequencing. There's a lot of smaller, more focused groups that we're working with, because it's not just about sample numbers. It's not just about sequencing millions of samples. It's about collecting samples that have associated phenotypes that are going to lead to actionable discovery, right? So, it's not, size matters, but it's not everything. I'll leave that at that. So thinking about how to do this, we want to move quickly. We're motivated to move quickly. And when I showed up at the RGC, I was sort of shocked because I hadn't seen an infrastructure that was able to move this quickly. An automated biobank, a library prep automation that handles over 500,000 samples a year, and an Illumina fleet that handles over 500,000 samples a year. We're doing whole exome sequencing, if you want to normalize your expectations there. But I recently went on the tour that is given by our sequencing head, John Overton, where he walks us through, how everything works in the lab. No one lets me in a wet lab. I can't go in there. I'm not, cap clia or anything. They would lose their heads. I'm bad in there. So I went on the tour for the first time to understand the sequencing. And this entire process can be monitored and run by one individual, okay? There's probably a whole really interesting talk about how you can support 500,000 exomes a year with a huge amount of infrastructure, very clever design, and one individual a year. But it's pretty impressive. But what really struck me is that for the first time, data generation, this is expensive data, right? Generating 500,000 samples a year as exomes is expensive. I don't know the number. They don't let me do any of the money stuff either. They really just put me in a corner and say, worry about computers. But data generation is not the bottleneck. For us, it's the driver. Sequencing is a complex, long process. There's lots of, I don't want to say manual anymore because it's all robots doing it, but it's not done in silico. There's lots of expensive things that go on. It's the bulk of the cost of doing of generating a sequence sample is doing the actual sequencing. So you'd think the most expensive step might be the bottleneck. It's not. We're generating data so fast that it's actually a challenge for me on the computational side to process, analyze, QC that at speed. So these are my challenges at speed. I have to keep up with the sequencing. It's not the other way around because we have all these samples coming in. So I've modified this slide a bit to go through, without offending anyone, what the challenges are. We have cohorts. We put about one freeze out a week, one deliverable to a partner. We have 500,000 samples a year, which works out to about a sample a minute that we're putting out for exomes. Our challenges there are maintaining 24/7 uptime. The sequencing pipeline very rarely goes down, for just sort of errors. Sometimes there are things that happen, but I have to make sure that I'm processing as fast as the data come off, because that's when you really lose money when you have one of these fleets operating capacity. If you're not running samples, then you're losing money. So I have to make sure I'm maintaining my uptime. Obviously, we have to do quality control and QA at scale, because when we're cranking out this many samples this fast, if it takes me a week to figure out a problem, that's a week of money spent that I have to go back and redo. Okay, so I have to figure out these problems fast. And then variants. I have an estimate here that I'll talk about in a minute that I have about one new variant a minute. And I have to figure out how to aggregate them. One new variant. That's one new variant that I haven't seen before every minute. And I'll walk through whether that number makes sense. So in order to have this at-speed infrastructure, our philosophy is to partner and to build as needed. And here is just sort of a general picture I have that details some of our partners, obviously including DNAnexus. What I want to draw attention to, I'm just going to go back and forth a bit. In terms of the data delivery and uptime, building an infrastructure where you can have 70+ collaborators and you need to be able to have tiered control for them and be able to move those files around without having a huge duplication of your footprint, and maintaining uptime on a cloud provider, that's really where the infrastructure of DNAnexus has been game-changing for us, okay? So I'll just sort of put that out there now that, a lot of people say, oh, well, you can run your pipeline on the cloud raw, or you can do whatever raw, but being able to do this at speed, at scale, robustly, with full uptime, and be able to sort of manage the security, the compliance, the access control, that's really where we've leveraged DNAnexus's expertise for those steps. This is the new slide. I always try to have one piece of new data that I show for every talk. This is the new one. This is sort of the growth of what variants we see. I'll try to use the mouse. Oh, I'll screw that up. So, this is our biggest cohort that we ever sequenced. It was about 150,000 samples. And, we saw, what is that, like 8 million variants. And then as we added our progressively larger cohorts, we can see how the number of variants that we see are growing. And for reference here, I have dbSNP. I think this is dbSNP version 150, 7.6 million variants. I don't know how many samples is dbSNP because I think you just submit variants. But then version 2.1 of Nomad exomes is 125,000 whole exomes. And there was about, I think, 16 million, 17, no, 11 million variants, 9 million variants just in the capture region that we're talking about here, which got covered up a little bit. But looking at this trend, we are seeing about if we just extrapolate, it's about 25 new variants per sample. Every time I do a cohort, I get about 25 variants that I don't already have in my internal database. And as you can see, just in our 39 meg capture region, we have 23 million variants. If we look at everything we see, everything that we call variants on, including a buffer, it's more than double that. And we can see that that's growing. So we're seeing a ton of variants that we have to sort of process as we go. And it's not just the variants that we see, it's also this genotype matrix that we have to process. I need to have a genotype for every variant in every sample. These are just a couple of different large cohorts that are out there. And as you can see, TopMed Freeze 5, which is what you would get if you went to the Bravo browser, the whole genome, 62,000 whole genomes with about 463 million variants. We have about as many genotype entries in our exomes at just about 500,000 as TopMed has in its 62,000 whole genomes. So thinking about how to manage and deal with that size of data and process that is one of the challenges. We partner with Databricks and have an entire team led by Lucas and Evan at the RGC, where they're using distributed compute infrastructure in order to be able to accelerate our queries to push this discovery faster. What you can't do is try to parse a VCF or a Plink file with that much data. You need to have something more clever, and we're doing something, for our queries, we have infrastructure for that. In order to aggregate these variants, we've worked closely for, years, I, in both of my gigs, work closely with DNAnexus on GLNexus, which is how we're aggregating these variants into large squared off project level VCFs. This is an example of doing about 240,000 whole exome samples. Currently, we are trying to break GLNexus by doing 633,000 whole exomes because we know that's what we're moving towards. So we're scale testing it now. So by the time we actually want to do a project of more than 500,000 individuals in one set, we have the infrastructure to do that. You have to be innovating in order to do this. There is no book for doing these types of analyses at these scales. We have to figure that out. We have to address what we think our data structure should be, what our protocol should be. What this allows us to do is at-speed discovery. The 50K project level VCF, we can do that in less than a week. That's going from, we have the data come off the machine, we process everything, and then we get this is from the preprint paper that's on bioRxiv that actually gives us our loss of function analysis. And there's other things that get done like the association test. We can crank through that in 1 week. That's what I mean by at speed, because we have these data coming off and we need to get the results fast. Why do we need to go fast? Because patients can't wait, like I said. There's no intrinsic bottlenecks. The data are the driver. The other thing is that operating at speed allows us to develop at scale. Recently, we had to update a 50K sample project level VCF. We updated our single sample, but we also updated our single sample variant calls, but we also updated our version of GLNexus. When we compared the old and the new, we changed two things, and we saw differences. And we said, well, we want to understand what the effect of each was. So instead of trying to do some clever downsampling or inference, we just ran the other two. We changed one thing and not the other. We changed one thing and not the other. And we were just making 50K sample project level VCFs just for the purpose of R&D. We were never going to put these out. We're just doing them for the R&D purposes. It took us about 2 days to make them. We did all the pairwise comparisons, and we're able to isolate these differences. So being able to operate at speed allows you to do your R&D at the scale of your research. You're not downsampling, you're not guessing, you're not inferring. So just as a summary, because I know I'm running out of time, when we think about operating at speed, these are some of the things that have come up as challenges, and the solutions for some of these, having a partner with DNAnexus really helps us manage those data, get them out, maintain uptime, give us that speed. To do quality control analysis and QC, that's our at-scale R&D. That's the ability just to do things quickly, cheaply, at a large scale, so we're not downsampling, we're not titrating, we're actually looking at the data and we're QCing it there. And then in order to aggregate and analyze these huge number of variants, the unprecedented number of variants, we have to partner with experts. GeoNexus, our friends at Databricks we've used in order to figure out what the innovation is going to look like to do something that no one's ever done before. So I think that's probably about my time. Yeah, I'm getting a very polite smile. I'm not allowed to put a lot of logos up, but I do want to put up key genomic resources that are just critical for us doing R&D and things at scale. I'm happy to talk about how we've used any of these. And with that, if there's time for questions, I'll take them. If not, I'll sit my ass down.
Thank you, Will.